Hello. Here I am again. I've got our next topic. This is going to be a brief overview of some very basic chemistry. I just want to review a few things. Most of you probably had a chemistry class at some point. I just want to make sure we're all on the same page, and we want to build on this later on in the semester. And so we're going to just talk very basic, atomic structure. We're going to talk about chemical reactions and energy, and a little bit of organic chemistry. Again, this is important because we're going to use this the rest of the semester. And the first thing that we need to address here is this is a biology class. And so why are we talking about chemistry, one of the first lectures, and here we are talking about chemistry? Well, everything is chemistry. I mean, everything that goes on is chemistry. And you must understand chemistry to be a good biologist. Living things are just a coordinated set of chemical reactions. Everything that goes on in your body is a chemical reaction at some level. Everything that goes on in the world is a chemical reaction at some level. So chemistry is everything. Of course, the basic building block in chemistry is the atom. And an atom is also an element. You put atoms together to make molecules. If you want information about those atoms, where do you look? Of course, you look on the periodic table, where you get all the information you need about each element. Now, what you'll find out in biology is that we don't use most of this table. There's only a handful of elements that make up 99.9% of the molecules we're going to talk about. And so just kind of pay attention to that as we get further into organic chemistry. Okay, so again, very basic stuff. What's the center of an atom? Of course, it's a nucleus. Now, one thing that we need to point out, we're going to talk about cells in a little bit, and cells also have a nucleus, right? So if I ask you, what's a nucleus? You should ask me, well, what are you talking about? Are you talking about an atom? Are you talking about a cell? But now we're talking about an atom. And so, of course, what's in the nucleus? What's it made of? Neutrons and protons. And we need to talk about the charge of each. What's the charge of each one? Of course, the protons have a positive charge. Neutrons are neutral. And then, what's orbiting the nucleus? There you've got electrons. And what's their charge? Of course, electrons are negative. And that's the basic structure of an atom. Now, when you look at any element, any atom, its charge is neutral in its lowest energy state, or in a typical state. And so that means the number of electrons equals the number of protons. So if I tell you the number of protons, you should be able to tell me the number of electrons. If we look up something in the periodic table tells you the number of protons. It also tells you the number of neutrons. And consequently, you can figure out the number of electrons. And when the electrons equal the protons, then those negative charges balance out the positive charges, and it has no net charge. And so here's a very simple basic diagram of a carbon atom. And in the nucleus, you've got the neutrons and the protons, and then you've got the electrons orbiting the nucleus. Now, you notice how they've drawn this here. That's one of the important things we'll talk about. The electrons don't just orbit in any old haphazard way around the nucleus. They orbit in different shells. And so here you see two shells. You see an inner shell and an outer shell. And electrons can't exist in between those shells. The electrons can jump from one shell to another, and then they can jump back, but they can't exist. They exist in these discrete locations or discrete distances from the nucleus. And so that's going to be important later on when we talk about metabolism and photosynthesis. The further out the shell, the higher its energy state, the more energy it has. And so for an electron to go from an inner shell to an outer shell, you have to add energy. And then if an electron jumps from an outer shell to an inner shell, it has to lose energy. And so that'll be an important concept later in the semester. So here again, figure from your book showing the same thing, showing the carbon nucleus and the 6 electrons orbiting in their discrete shells. And since you've got 6 electrons here, you know that carbon has 6 protons. And it also has 6 neutrons, it turns out. And so you might recall that those electrons in the outer shell are called valence electrons, and that's called the valence shell. And that's sort of where chemical reactions take place. And atoms like to react, or atoms want to fill their outer shell, or to have their valence shell filled. I say want, right? Obviously they don't want that. But what that means is it's a low energy state. And so we'll talk about energy states a lot, but things that are at a high energy state spontaneously want to go to a low energy state, right? And so a ball on top of a hill will eventually roll down. If you push it, it's going to roll down. A ball at the bottom of a hill is never going to spontaneously roll up. You got to put energy in it to roll up. But that ball at the top of the hill already has a lot of potential energy and wants to roll down. Things want to go from a high energy state to a low energy state. Well, when this valence shell is filled, that's a low energy state. And so atoms will react with other atoms to try to get their valence shell filled because that's a low energy state. And so by knowing how many electrons are in that outer shell, that can tell you something about how reactive the atom is. And so different shells have different amounts of electrons, need different amount of electrons to fill them. That inner shell only needs 2. The next shell that you see here, it's got 4, but it wants 8. And then you'll learn your chemistry class, as you get different kinds of shells, they have different numbers of valence electrons. But here in biology, typically the stuff we work with is going to want 8 electrons in its outer shell. But that's important for chemical reactions. And so, like I said, for carbon, it wants 4 more electrons. It wants to have a total of 8. Okay? Well, it's not just going to go grab electrons, because then it would have a net negative charge, and that's not going to be at a high energy state. That's very unlikely to happen. So what's an easier way to get electrons without changing the total charge? Well, it's to react with other atoms and to share their electrons. And so that other atom is neutral, so you're not changing the overall charge, but you're getting that electron that you want to help fill out your outer valence shell. And so that's how we put a bunch of atoms together to get a molecule. And to get them to stick together, they share their electrons. And so it's that shared electron that binds the 2 atoms together, and we call that a covalent bond. And so you see there's that valent or valence word again, and co kind of meaning shared together, shared valence electron. And so this is what binds atoms together. So let's look at an example of this. So here's a different atom. This is a hydrogen atom. And of course, it's the most simple atom. It's just 1 proton and 1 electron, and that's it. And we'll talk about hydrogen and protons quite a bit in this class because they're so simple, they're so common, and they have a big effect on lots of chemistry. And so, for example, you're familiar with the concept of pH, right? Well, pH is measuring the concentration of these hydrogen ions. And that's because, again, they're so common and they're very reactive. So if you've got just 1 proton, that's a hydrogen ion. That's a hydrogen that's lost its electron. And so it has a positive charge. Well, it's a very reactive atom. And so they play a big part in lots of chemistry and in lots of things. And so we like to measure them, and a way we can measure the amount of those is with the pH scale. Anyway, this is a hydrogen atom. So it has a proton and electron. It's not an ion. It does not have a charge. This is just a regular old hydrogen atom. Okay. And so the carbon wants 4 electrons. Each hydrogen has 1 electron, so why not grab 4 hydrogens and share their electrons? The hydrogen still gets to keep its electron, so it's happy. The carbon now has a full outer shell, so it's at a low energy state, so it's happy. And so everybody's happy. And this is one way we can build a molecule. And so those electrons that originally belonged to the hydrogens are now shared between the carbons and the hydrogens. And so what happens is that electron, it's orbiting the hydrogen nucleus, and then when that hydrogen binds to the carbon, then that electron will kind of go around the hydrogen, then it'll go around the carbon, then it'll go around the hydrogen, then it'll go around the carbon. And it'll just kind of keep going back and forth. It'll orbit both nuclei, and in that way it holds those nuclei together. That's a covalent bond. And so what we just did is built a very simple molecule. This is a methane molecule, CH4. And so what we're showing you in this table here is there's all kinds of different ways that we can indicate different molecules. And some you'll see more often than others in this class. In your chemistry class, you might see things one way, and in here you'll see them a different way. The most common would be like CH4 or the structural formula. That's what you're going to see in this class most of the time. But you can also see the three-dimensional aspect in the ball and stick model or the space filling model and so on. And so these are all different ways we can represent the same molecule. Now when we look at that structural formula, like I said, this is how we're going to draw a lot of molecules in this class. And those lines that connect the different atoms, those represent covalent bonds. And so you can see you've got on each hydrogen, you've got a line to the carbon, and that means there's a covalent bond, there's a shared electron between those two atoms. And sometimes you can have more than one shared bond. You can have a double bond, and so there'll be 2 lines. And every once in a while you can share 3 electrons, and there'll be 3 lines. So on. Well, not so on, that's as far as it goes. Now we just made a very simple molecule by combining carbon with hydrogen, but of course carbon can react with lots of other atoms like oxygen or nitrogen or phosphorus. And you can just keep hooking together more and more of these atoms to make larger and larger molecules. And that's another thing that you'll find out when you look at organic chemistry or in biology, is biological molecules tend to be very big. Sometimes called the macromolecules. But it doesn't have to be just carbon and hydrogen. It could be lots of different things combined in different ways to make all these different molecules. And so here again is a figure from your book showing that methane molecule and showing those electrons being shared now. So if you look at carbon now, it has 8 in its outer shell. And each hydrogen, it kind of looks here like each hydrogen has 2 electrons, but it really just has that one original one, right? But that's covalent bonding. Now, again, one of the things we want to bring out here, we talk about, this is a science class, we talk about the scientific method, and a criticism some people might throw out is, well, you can't see these things, and so therefore, you're just guessing. They can't really, you can't really study something that you can't see. And that's not really true. You can't see an atom or a molecule, even if you look under a microscope. This is beyond the ability for us to actually see pictures of. Maybe very, very large macromolecules with an electron microscope you might be able to see. But in general, you can't see these things. But you can still create hypotheses which still allows you to make predictions, and you can test those predictions. And so you don't have to directly observe something to be able to study it in science. You have to be able to make predictions and test those predictions. And this is a perfect example, right? You can't see an atom or a molecule, but we know a lot about chemistry. I mean, just look around you. There's just, like I said, everything is chemistry. And if you've had a chemistry class, you realize we know a lot. I mean, we build atomic bombs, right? We figured out how to split atoms and release ridiculous amounts of energy. You can't do that by accident. And so how do we know so much about chemistry? By using the scientific method, right? Hypotheses are falsifiable, testable, and they make predictions. And so when you have an idea, when we're starting to learn about chemistry, and chemists and physicists had ideas about the structure of the atom, they could make predictions, and then they could test those predictions, and through that process learned more and more about the structure of the atom. So again, the important thing is that you can come up with a testable hypothesis that makes predictions. You don't have to actually observe something to learn about it in science. And so, studying chemistry is a perfect example of that. You can make predictions, you can test the predictions, and reject or fail to reject the hypothesis. And just like anything else, you go down a path and you think you have a good idea of the structure of an atom, and then some new data suggests that that's not a perfect model, and so then somebody comes up with a different model, and over time you learn more and more. So anyway, it's a perfect example of the scientific method working and how it works. Okay, so let's talk about chemical reactions and energy a little bit. And so when we make these molecules and we bring these atoms together, that doesn't usually happen spontaneously. It takes energy to join these atoms. You're going to have some reactions that occur spontaneously, but in biology, to build organisms and to build these biological molecules, that's not going to happen spontaneously. You need to use energy. Of course, this is why, you have to eat 3 meals a day, or if you're a plant, you need sunlight, or all living things need to take in energy and transform energy, and this is what they do with that energy, is they build molecules. And so big picture, all that energy that's in living things started in the sun. The sun has fission, and there are nuclear reactions that release energy, and that energy is in the form of heat and light. And so then that energy travels through space and hits our planet, and then that light energy gets captured and turned into chemical energy. And that's big picture, that's the flow of energy to living things. It starts with nuclear energy in the sun, converts to light energy, and then gets captured into chemical energy. And so that's what this figure is trying to show you here, is that sunlight is energy that reaches us from the sun, obviously. And primary producers, photosynthetic organisms, have the ability to capture that light and to use energy from that light to build these organic molecules. And then other herbivores eat those plants and take in the energy from those plants, and then carnivores eat the herbivores, and that energy gets passed from the trophic levels. And so that light energy, when it gets stored in that molecule, becomes chemical energy. And then that chemical energy gets passed from organism to organism until it's eventually all turned into heat. And so let's talk about, just very, very broadly, how does that energy get stored in a molecule? What is chemical energy? of course, if you in your physics class and your chemistry class, you talk about this in a lot more detail. We just want to have kind of a conceptual model of this to help us work through the rest of the semester. So here's a new molecule. This is a urea molecule. It's a little bit more complicated than our methane molecule. You see we've got some nitrogens in here. You see that double bond between the carbon and the oxygen that I talked about. So that's 2 shared electrons. And the double bond is much stronger than a single bond, and the triple bond is crazy strong. Anyway, well, where do you think, we find these urea molecules? Maybe you've heard of urea. Where do you think we find it? Do you find it on Uranus? Do you find it in urine? Do you find it in shark blood? Or do you find it in skin cells? There's actually 2 answers are correct here. You find it in urine, and it's an important way that we get rid of nitrogenous wastes. But you also find it in shark blood. And so sharks use this to help them with osmoregulation. And we'll talk about osmoregulation, but basically, sharks live in salt water. And so their external environment, the water they live in, is much saltier than their blood. And consequently, that means that they're constantly losing water to their environment. And by retaining urea in their blood, they help to reduce that salt difference. And so they don't lose as much water as they would. That's osmoregulation, and we'll talk about that. Okay, so how did we get all these atoms to come together, right? So we had to have the carbon and the oxygen, a couple nitrogens, a couple hydrogens, all had to come together and start sharing those electrons. And like I said, that doesn't happen spontaneously. We have to put energy in. Those molecules have to be, those atoms have to be brought in proximity so that then they can start sharing that electron. And that doesn't really happen very much spontaneously. You have to put energy in to bring those atoms together. And so when you bring them together, initially they want to repel each other. Because really, what are you talking about when you've got these atoms? They've got this nucleus and they've got these electrons, right? But there's a lot of space around those electrons. But that nucleus is very dense. And you've got protons and neutrons. So those protons have positive charge. So you've got a dense cluster of positive charge. Now you've got those negative charges flowing around, but they're not as dense as the positive charges in the nucleus. And now I'm bringing another atom and another nucleus together. Well, what happens? You've got positive charges and positive charges. What do similar charges do? They repel each other, right? They don't want to come together. And so as you bring them close together, they're going to repel each other. So to get them closer together, you have to do things to actually put energy in to bring them close together. When you bring them close enough, then they can start sharing that electron. And when they start sharing that electron, then that binds them together. And so they don't, those nuclei want to push apart because of their similar charge, but because you've got this negative charge that's orbiting both of them, that's sort of canceling out those positive charges, and it's bringing everything to a lower energy state, that holds them together. The point is it took energy to start this. It took energy to get them close together. And then once they started sharing the electron, then they'll stay together. But we have something called the first law of thermodynamics, which is that energy is neither created nor destroyed. It just changes forms. And so when you put that energy in, that energy has to go somewhere. It has to, the energy that goes into making this can't just disappear. Well, it gets stored in that molecule. And that's what I mean by chemical energy. And so that energy is in the bonds of that molecule. And if you rip those atoms apart, again, that energy has to go somewhere, and that energy can be released. But if you do that in a controlled environment, if you do that in a certain way, that you rip them apart, you can use that energy. When that energy is released, you can use that energy to do something. And that's what living things do. And so those lines represent covalent bonds, shared electrons. If you rip this molecule apart, the energy that's stored in those bonds is released and can be used for something else. Now we have another law called the second law of thermodynamics, which says that all energy transfers are inefficient. And so every time you change energy from one form to another, or you do something like this where you're, putting energy in and releasing energy, a little bit of that energy just gets turned into heat. Doesn't get stored in the bond. It just goes to heat. And eventually all energy is going to go to heat. That's the thermal death of the universe, and that's a total bummer for everybody. But it won't be for a few years. And so when we break those bonds, we release that energy. A little bit of that energy turns to heat, but the rest of that energy, if we're smart, we can capture it and put it into a different molecule, or we can do something with it. We can do a different chemical reaction. Well, ultimately, that is what life is. That's what living things are doing. They're taking molecules from their food, ripping them apart, taking the energy that's released and using that energy to do other things, to either build new molecules so you can grow, or to do other chemical reactions, to make you run, or to make your heart beat, or whatever. And so that's what I'm saying when I say that living things are just coordinated chemical reactions. That's what I'm talking about. That's really all it is. And so ripping apart molecules to release energy, then using that energy in other chemical reactions, that's what living things do. And that's kind of one of the things that helps to define what is a living thing. And so to kind of help explain this a little bit, I've got my famous tennis ball metaphor to help you kind of help explain this idea of energy being stored in covalent bonds. Okay, so pretend you've got two tennis balls and a spring in between them, all right? And so then you push the tennis balls toward each other and compressed that spring. So then what would happen if you let go of the tennis balls? Well, they would go flying in each direction. But why? What causes them to fly in each direction? Well, you compressed that spring, and so then when you let go, that spring pushes them in either direction, right? So you stored energy in the spring. When they're just sitting there attached to the spring and it's not compressed, nothing happens. But you compressed them. You put energy in, and that energy gets stored in the spring. And so then when you let go of the tennis balls, the energy in that spring pushes on them and causes them to fly apart. Well, what if when you push the tennis balls together, then you had somebody come and wrap duct tape around them? Now would the tennis balls fly apart? They want to fly apart, but they can't because the duct tape is holding them together, right? And so there's energy stored in the spring between those balls, but the duct tape's holding them together. And so this is the metaphor. The tennis balls are like atoms, and the duct tape is the bond holding them together. And the energy in the spring is the energy that's in that bond. And so if you break that bond, if you cut that duct tape, now that energy can be released. But as long as that bond is there, that energy is being stored. And so as long as the bond holds the atoms together, energy is stored in the molecule. So this is the metaphor, right? But it's the same thing with metaphors, with molecules in biology, is that when those atoms get held together, there's energy in there. But if you rip them apart, that energy gets released. And you can use that energy to do something or to do other chemical reactions. And so this is big picture how the whole system works, right? As long as the sun is burning, and as long as light is hitting this planet, and as long as there's photosynthetic organisms that can capture that light energy and turn it into chemical energy, then that chemical energy can get passed to all the other living things, right? Without those photosynthetic organisms, then none of the rest of this works. And so that's how living things can be alive, because some organism somewhere captured that light energy and turned it into chemical energy. But as I mentioned, every time you rip the molecules apart and you use that energy, you lose a little bit to heat. And so that energy that gets captured by the sun, it gets passed among all these organisms, but each time you lose a little to heat, you lose a little to heat, and eventually it's all gone. And so you need that sun to keep sending light this way, and organisms need to keep eating because they keep needing that energy, and when you lose the ability to harvest that energy, that's it. That's when you're dead. So again, here in my metaphor, if I cut the tape, the tennis balls go flying off. And so the energy that was stored between the atoms is released when you break that bond. So it's a metaphor, okay? It's just to help you kind of visualize what's going on here. So chemistry majors, physics majors. Spare your comments. You know, I'm just trying to just help us all understand this in a little bit. I know it's not exactly the way it works, but it's a pretty good metaphor. And so, like I said, that's what life is. Life is harvesting energy from other organisms, ripping apart the molecules, using the energy that's released to do other chemical reactions that you need to stay alive. And so all living things are made of biological molecules, and there's energy stored in the bonds of those molecules. And when you lose the ability to harvest that energy, or the ability to put that energy to good use, well, that's when you die. And so living things take in these molecules, rip them apart to harvest the energy, and then use the energy to make new molecules. Well, where does all this energy ultimately come from? What starts the process? Well, like I said, the plants capture the sunlight energy and use that energy to build plant molecules. So plants aren't out going and eating other organisms. Yes, you've got the Venus flytrap, and you have some very interesting carnivorous plants, but they get some energy, but they get a lot of essential nutrients from those flies they eat. I think they still photosynthesize some. But the plants are able to take that, or photosynthetic organisms, doesn't have to be a plant, is able to take that light energy and store it as chemical energy in their molecules. And then herbivores come along, eat the plant, rips apart those molecules, and then the energy is released. Then the herbivore captures that energy to make their own molecules, and also to do other reactions, again, to make like your heart beat, to make your nerves work, to be able to walk around. And so there's lots of chemical reactions that need to take place to make this herbivore live. And those all take energy, and this is where the energy comes from. And then energy is stored in the herbivore's molecules, which get eaten by a carnivore, and they do the same thing, and so on, until all that energy gets turned to heat. Okay, so again, I said this was going to be very basic chemistry, just to help us kind of wrap our mind around things as we go through the rest of the semester. Let's talk a little bit about organic chemistry. And so, when we talk about organic chemistry, of course we're talking about chemical reactions in living things, but a real good definition is organic chemistry is the chemistry of carbon. So living things are made of carbon. So all our biological molecules throughout every cell of your body, of every living thing on the planet, is mostly made out of carbon. So that's why I talked about carbon at the start of this whole thing, because carbon is the essential atom of living things. And so organic chemistry is the chemistry of carbon. So as we talk about these molecules, note that they're all based on carbon. And in biology, we get these carbon chains. And so you tend to get long stretches of carbons, a bunch of carbons stuck together in different ways. And they can vary in length and they can vary in shape. And that's how you get different organic molecules. And so here's, for example, a couple of different simple organic molecules that vary in length. We started with a methane, which was a C and 4 H's. Then you've got ethane, which is CH6. And then you add another carbon and 3 more hydrogens, and you get propane. So by just making it a little bit longer, you get a different molecule. But you can also just arrange the atoms differently and get a different molecule. So here you see you've got the same number of carbons and the same number of hydrogens in each of these molecules, but they're arranged a little bit differently. But that gives you a completely different molecule. Remember, we talked about double bonds. And you can have double bonds, and you can have double bonds in different places. So here again, you've got the exact same number of carbons and hydrogens, but that double bond is in a different spot. Gives you a different molecule. One of the things you'll find out when you start to study organic chemistry is that carbon likes to form rings. And so we've been drawing this as you kind of got like, a long line of carbons. But when you get 5 carbons or 6 carbons, that string of carbons tends to wrap around and connect to itself and form a ring. And so that's what they're kind of showing you here. They're showing you a couple of different molecules that have, both of these have 6 carbons and a few hydrogens. But instead of being, now these 6 carbons could be in a linear form. They'll act like that. But when they bend around and form a ring, that sometimes is a lower energy state. And so this is how we represent it. And so you can see we've got the carbons and hydrogens drawn here, but then you also got like a little hexagon drawn. And so whenever you see a ring like that, that's what that means. It's just a ring of carbon. And usually these rings in biology, they're 5 or 6 carbons. That's a nice low energy state. If you have more carbons, they don't really form a good ring. If you have fewer carbons, they don't form a good ring. And so this is, again, just something that you're going to see throughout the semester. And so usually we draw the rings like I've indicated here. And so each straight part indicates a covalent bond between carbons. And so if you look on the right, you see there's a couple places where we've got 2 straight lines parallel to each other, and then those represent double bonds. And then you see in the simplified form that's indicated here, you don't see any hydrogens. That's because hydrogens are assumed. Again, I told you hydrogens are everywhere. And hydrogens take place in all kinds of reactions. And so it's not worth drawing every hydrogen, because if there's an empty spot, if a carbon needs one more electron, it just grabs a hydrogen. And so we just assume it. So in these rings, every vertex, every point represents a carbon, and then there's enough hydrogens hanging off that carbon to fill out its shell. So this is just a simpler way. So again, when you see these rings drawn, realize that you're looking at carbons and hydrogens in that ring. And so here's a figure from your book, again, showing you some rings and showing you how now, once you have these rings, now the rings can get stuck together and you can form a long line of these rings, and you make a lot of different biological molecules that way. And you can see here that you've got is that a 5 carbon? No, they're both 6 carbon sugars. But you've got a couple of rings here. One is a glucose, one is a fructose, and you can see that they have, somewhat similar organization, but they're organized a little bit differently, and that makes them a different molecule. And then those two get joined together to form a sucrose molecule. So in the next chapter, we'll talk more about biological molecules, but what you've got here are monomers, the building blocks, and then you bring those together and they form polymers, or the big macromolecules. And so then in the bottom part of this, you see, again, you've got some different 6 carbon rings, and they're attached together in a certain way, and that's how you make cellulose, which is, a very common carbohydrate, and it makes up the cell wall of plants and things. Now again, whenever we look at these molecules, don't forget, every one of those covalent bonds contains energy. And so if you break that bond, you release the energy. And so the more bonds you see, the more energy that molecule can contain. And so if you look here, which of these molecules then would have the most energy based upon what I just said? Well, it's probably going to be B. Because look at that. That's got lots of carbons and lots of hydrogens, and they're all connected with those bonds, and every one of those bonds has energy. And that's actually what we call a hydrocarbon, right? So it's not a very creative name, hydrogens and carbons together. But you get lots of hydrogens and carbons together, you got lots of bonds holding them together, those bonds have lots of energy. Well, what's a typical hydrocarbon? You've heard the term hydrocarbon. When you think of hydrocarbons, what do you think of? You think of gas and oil and fossil fuels, right? Well, what are fossil fuels? First off, what do we do with fossil fuels? We burn them to get their energy to do things. Well, why do we use fossil fuels and not other kinds of molecules? Because fossil fuels are hydrocarbons. They've got lots and lots and lots of bonds, which means they've got lots and lots and lots of energy. And so you can get a very high energy density. You get lots of energy in a small package, and that's why we use fossil fuels. And again, what are fossil fuels? They're formerly living things that got buried 100 million years ago, and they just got pressed deeper and deeper, deeper in the earth, and more pressure. And so you just pack all those organic molecules tighter and tighter and tighter, and so you pack all that energy into a smaller and smaller molecule. And so when you dig it up, you've got a molecule that has lots of energy in it. At the same time, when you burn that, you're also releasing that carbon that's been buried for 100 million years. And that's a big problem, what we're going to talk about. And so again, this is a hydrocarbon, just hydrogen and carbon, nothing else. So that's kind of the first macromolecule we're going to talk about. That's what a molecule of gasoline looks like, or a molecule of oil. It looks something like this, but very, very long. Lots and lots of carbons and hydrogens and lots of energy. Now, here's a theme that I'm going to be coming back to, and I'm going to emphasize again and again. I told you that just slightly rearranging the molecules or moving the bond gives you a completely different molecule. And to me, that's one of the fascinating things about all of this, is that a very slight difference at the molecular level can have a huge difference in living organisms. And so, again, we've already seen this, a very slight difference, just rearranging the carbons a little, or just moving a double bond, gives you a whole different molecule. And that whole different molecule has very different properties. So very small changes at the molecular level are very important at the organismal level, at the biologic level. So that's another reason why we start with chemistry, and we have to talk about chemistry. Because if you want to understand what's going on in biology, you got to understand these very subtle differences at the molecular level. That helps to explain a lot. So here's another example. Here's two complex biological molecules. And you see that we've got those ring structures, and we've got several rings together. And both of these have six carbon rings, and then one 5 carbon ring, kind of all stuck together. And so it's again, biological molecules tend to be big with lots of things stuck together. And this is what we're talking about here. So we've got two molecules here, and they have very similar structure. Look at the shape of those hexagons and that pentagon. Look at where the bonds are. You've got the methyl group, this CH3. You've got a hydroxyl group, this OH. They're very similar, but they're also just a little bit different, right? And so they're two very different molecules that have two very, very different effects when you talk about the organismal level. So I've got the names here. What are these? One's estradiol, one's testosterone. What are those? Well, those are, female sex hormone and the male sex hormone, right? And so depending upon where you find those hormones in what cells and in what body, they have very different effects on the organism. They're very similar. There's only a little bit of difference at the molecular level, but it's a huge difference between the female sex hormone and the male sex hormone. That's the point I'm trying to make. You're going to see this over and over again, that it takes a very small change at the molecular level to have a big change at the organismal level. So that's why we need to talk about chemistry a little bit. And hey, that's what I just said right there. Big changes at the organismal level. Okay, so I said we were just going to have a brief overview of chemistry. That's it, right? That's the basic ideas that all we really need to understand what we're going to talk about the rest of the semester. There will be times where we're going to dig a little deeper into the chemistry, but basically that's all you really need to know to understand a lot of the things that we're going to talk about. Now, the more chemistry you know, the better you understand biology. And so in the next lecture, we're going to talk more about organic chemistry, biological molecules, but then that'll really help us segue into the rest of the semester. So that's basic chemistry. That's really all I'm going to talk about right now. Let me know if you've got any questions, and I will see you later.